Good morning, students. So in this class we will continue our chapter 2, that is about the histological organization of plants. So in this histological organization of plants, previously we have discussed about how plants have been organized. ఎలా ఒక plant లో root system అనేది ఎలా develop అయింది, and what are the inside tissues, what are the inside characters that are present in that type of a root system. అలాగే స్టెమ్ ఎలా డెవలప్ అయింది, స్టెమ్ లోపల ఉన్న టిష్యూస్ ఏంటివి, సెల్స్ ఏంటివి. These are all a part of this histological organization of the plants. This is also called as internal organization of the plants. How plants are being organized internally. For example, if you take human body consists of various types of organs. Human body lo chala rakamaina different types of organs అనేవి ఉంటాయి. So how that organs have been arranged inside. Organs అని ఎలా arrange అంటే heart will be on our left side, our stomach will be on our right side, pancreas ఉంటాయి, liver ఉంటుంది, digestive system ఉంటుంది. Like they are be arranged in a systematic manner. In a similar way, how the plant internal parts have been arranged. Plant లోపల ఉన్న cells అయి ఉండొచ్చు, tissue అయి ఉండొచ్చు. How are they arranged? That all we are reading in this chapter. So previously we have described about the different types of tissues as well as the tissue systems, and especially or particularly in the yesterday's class we have done with our transverse section of a primary monocot root as well as a dicot root. Monocot ante enti single cotyledon unnadi, dicot ante di is nothing but a two, so two cotyledons unnadi, and we did a transverse section of both monocot root as well as the dicot root. మనకి monocot కి సంబంధించిన రూట్ ఇస్తారు, dicot కి సంబంధించిన రూట్ ఇస్తారు. సో అప్పుడు దాన్ని ఏం చేయాలి అంటే that are being already preserved under some sort of a solution or some sort of a watery content, అంటే ఒక fluid లాంటి content లో వాటిని స్టోర్ చేసి ఉంచడం జరుగుతుంది. And when we are doing our experiment or when we are doing our practicals, they will be giving that small sort of a root into our hands. So when we take that root, it will be very minute in its structure, చాలా చిన్నగా ఉంటుంది. దాన్ని తీసుకొని in a lab we will be having some watch glasses ఉంటాయి. So watch glass is like somewhat a glass shaped, అది గ్లాస్తో మేడప్ అయి ఉంటుంది. దానికి కొంచెం సన్నగా చిన్న ఇలా లొందలాగా ఉంటుంది. అందులో కొన్ని వాటర్ యాడ్ చేసి ఈ యొక్క డైకాట్ రూట్ అయి ఉండొచ్చు లేదా మోనోకాట్ రూట్ అయి ఉండొచ్చు, ఆ రెండింటిని అందులో ప్లేస్ చేయడం జరుగుతుంది. After placing that monocot root or dicot root into that watch glass, what happens exactly? You are placed, you have taken that out, దాన్ని బయటకు తీసుకుంటాము. ఆ వాటర్ను తీసి you need to hold that with our two fingers, only these two fingers, our thumb and our forefinger. So దాన్ని తీసుకొని you need to take a very sharp blade తీసుకోవాలి. That is a new blade, it will be so sharp. So after taking that blade, if you need to catch the blade like this, you need to hold the dicot root or monocot root with your two fingers and then hold it and then make it a slices. Cut it and make it into a thin slices. If you remember, there will be some sort of cheese slices and ఎంత థిన్గా ఉంటాయి. సో మీరు కట్టింగ్ ఆ ట్రాన్స్ఫర్ సెక్షన్ అనేది ఎంత థిన్గా చేస్తే మనకి అందులో ఉన్న టిష్యూస్ అంత విజిబుల్ అవుతాయి, అంతగా క్లియర్గా కనిపించడం జరుగుతుంది. So after slicing it, after making that dicot root or monocot root into your slices, dip that blade into a water. So that ఏమవుతుంది, ఆ బ్లేడ్ పైన అతుక్కొని ఉన్న ఆ యొక్క స్లైసెస్, ఆ రూట్ యొక్క కట్ చేసిన పీసెస్ ఆర్ స్లైసెస్ ఏం చేస్తాయి అంటే వాటర్లో తేలుతూ ఉంటాయి. ఆ వాచ్ గ్లాస్లో ఉన్న వాటర్లో మనం ఆ బ్లేడ్ని డిప్ చేసినప్పుడు ఆ వాటర్లో తేలుతుంది. ఆఫ్టర్ దట్ యూ నీడ్ టు యాడ్ సమ్ స్టెయిన్. సో దానికి స్టెయిన్ యాడ్ చేస్తాం. స్టెయిన్ యాడ్ చేస్తే ఏమవుతుంది, స్టెయిన్ ఈజ్ నథింగ్ బట్ ఏ కలర్. సో ఈ టిష్యూ నార్మల్గా వైట్ కలర్లో ఉంటుంది. సో ఆఫ్టర్ యాడింగ్ one drop of a stain to that, this tissue absorb the stain from that water. ఆ స్టెయిన్ని అబ్జర్వ్ చేసుకొని అది ఒక కలర్ని చూపించడం జరుగుతుంది. ఆఫ్టర్ దట్ by using a small painting brush that is present in our dissection box, by taking that brush you need to move on one thin slice. ఏదైనా ఒక థిన్ స్లైస్ని సెలెక్ట్ చేసుకొని దాన్ని కాస్త దీనిపైకి మూవ్ చేయాలి, స్లైడ్ పైకి. మూవ్ చేశాక దాన్ని మళ్ళీ మనం చేయితో టచ్ చేయకూడదు, బికాజ్ ఎనీ టిష్యూ విల్ బి డ్యామేజ్డ్. అది టిష్యూ కాబట్టి అందులో ఉన్న సెల్స్ కానీ టిష్యూ అనేది డ్యామేజ్ అవ్వడం జరుగుతుంది. సో బికాస్ ఆఫ్ దాట్, టు అవాయిడ్ ద డ్యామేజ్ యూ నీడ్ టు బి స్లైట్లీ టచ్ ఇట్ విత్ అ బ్రష్ అండ్ పుట్ ఇట్ ఆన్ టు ద slide. After keeping that onto the slider, next take a cover slip. Cover slip అనేది చిన్న గ్లాస్ లాగా ఉంటుంది. అంటే దాని ఈ స్లైడ్ పైన మనం ఎప్పుడైతే ఆ యొక్క ట్రాన్స్ఫర్ సెక్షన్ ఆఫ్ రూట్ పెట్టినామో దానిపైన ఇది ఇలా వేసేయాలి. వేసినప్పుడు మనం దాని ఇలా పైన జస్ట్ యూ నీడ్ టు ట్యాప్ ఇట్ సో దట్ that slice is attached to the cover slip. When we tap that, the slice is attached to that cover slip. And remember one thing, when you are putting a cover slip onto the slider, when you are putting a cover slip or when you are adding the cover slip onto the slider, remember that to avoid air spaces అనేటువంటివి రాకుండా ఉండాలి, because the air should not go inside that cover slip or the air should not go in between those cover slip as well as the slider. Now cover slip is being placed. Now go and watch that slider under the microscope, adjusting the light under 10x. So when we are observing, we'll be able to recognize what do you mean by actually that root transverse section. Any layers ఉన్నాయి, దేంతో మేడప్ అయింది, ఎపిడర్మిస్ లేయర్ ఉందా, కార్టెక్స్ లేయర్ ఉందా, స్టీల్ లేయర్ ఉందా అనేది తెలుస్తుంది. So as far as yesterday's class is concerned, we have discussed about the dicot root as well as the monocot root. Both will show the similar characters like after the transverse section. Both will show the similar characters. ఏంటి, ఎపిడర్మిస్ అనేది ఉంటుంది. ఏముంటుంది, there will be a presence of epidermis. Epidermis is nothing but an outermost layer ఉంటుంది, అంటే బయటికి ఉన్న లేయర్ అనేది ఉంటుంది. And that epidermis layer is protected by a waxy coating layer called as a cuticle. And from that epidermis there will be a protuberance that will protrude outside of that. And hair like projections ఉంటాయి, వాటిని మనం epidermal hairs అని అంటున్నాం. And this epidermal, the main function of this epidermis is it will provide some protection to the internal structures or internal tissues of the plant parts. Okay, and next if you move on to the second most important thing, that is the cortex. And cortex involves three types of structures, that is exodermis, general cortex and endodermis. Cortex lo only three types of structures ఉంటాయి, exodermis, endodermis and general cortex. Okay, so exodermis is the outermost. It is made up of 2 to 3 layers of parenchymatous cells, which are thin walled parenchymatous cells. And our parenchymatous cells అయినప్పటికీ ఏమవుతుంది, ఒక సెల్కి ఇంకో సెల్స్కి మధ్యలో there will be a presence of intracellular spaces. ఒక సెల్కి ఇంకో సెల్కి intracellular spaces అనేటువంటివి ఉండడం జరుగుతుంది. ఓకే, so this is all about the exodermis. Coming to general cortex. General cortex లో cortex layer cells ఉంటాయి, 3 to 6 layers లాగా అరేంజ్ అయి ఉంటాయి. అండ్ ఎండోడర్మిస్ వచ్చేసరికి ఆ స్టీల్ ఏదైతే పార్ట్ ఉందో దాన్ని స్టీల్ని కవర్ చేసే పార్ట్ని మనం ఎండోడర్మిస్ అని అంటాం. And when we are moving into the steel, this steel consists of a vascular bundles, and in case of this dicot and monocot root, vascular bundles are exarch in condition. Exarch ante protoxylem is facing towards periphery, metaxylem is facing towards center, that is towards inner side. Okay, so the vascular bundles are radial and separate, exarch, and it can be the cambium is absent. Because of the cambium is absent, they are said to be closed. Okay, and next if you see about the dicot root lo kuda same untundi, and there are some sort of differences between a monocot root as well as the dicot root. And if you see the major differences, TS of monocot root, TS of dicot root. If you see the major differences, this dicot root lo ఉన్న pericycle అనేది lateral branches ki rise istundi and vascular cambium ki kuda rise istundi. It gives rise to vascular cambium as well as the lateral roots or lateral branches. Okay, and when coming to this, this is a dicot. Okay. So next, when coming to this monopod root, this monopod root gives monopod roots lo una pericycle kasta, it gives rise to only lateral roots. It gives rise only to the lateral roots. And next, if you take the vascular bundles, here the vascular bundles in dicot roots are radial and separate, closed, and the xylem is diarch, triarch, and tetrarch. Diarch contain 2, triarch contain 3, tetrarch contain 4. And in this, the vascular bundles are only polyarch. So in this case, the vascular bundles are only polyarch. And coming to the medulla and pith, and next in this, the medulla and pith is completely reduced or absent. Indulo medulla anedi completely reduced or absent untundi. And if you take so if you move on with this, the medulla and pith is completely absent. Here it is well developed. So these are the main differences between a transverse section of a dicot root as well as the monopod root. And now we will see the transverse section of a dicot stem as well as the monopod stem. Root అయిపోయింది కదా. Now we will see about the transverse section. Dicot stem and monopod stem. So before drawing diagram or before showing diagram, first I will discuss even this dicot stem as well as monopod stem also consists of 3 parts of structures. 3 parts again, that is an epidermis, cortex and stem. Similar way lo indulo kuda 3 parts e untayi. Enti avi? First one vachesi epidermis, second one vachesi cortex, third one వచ్చేసరికి stem. And coming to the epidermis ఎప్పటికైనా ఇది గుర్తుపెట్టుకోవాల్సింది, epidermis consists of an outermost layer. It is an outermost uniseriate layer. Okay, it is outermost uniseriate layer which consisting of rectangular, tubular, compactly arranged cells. అంటే ఒకదాని తర్వాత ఒకటి సెల్స్ అన్ని కూడా కాంపాక్ట్ గా అరేంజ్ అయి ఉంటాయి ఆ యొక్క లేయర్ లో. And when coming to the function of this epidermis provide protection. ఈ ఎపిడర్మిస్ అనేది ప్రొటెక్షన్ ని ప్రొవైడ్ చేస్తుంది. And it is covered by some waxy coating substance called as a cuticle, and that cuticle will be helpful for the rate of transpiration. The rate of transpiration ki help అవుతుంది. And next, if you move on to the cortex, and now I am discussing about this dicot stem here. This dicot stem is also differentiated into 3 types: epidermis, cortex, stem. So if you move on with this epidermis emo already manadi అయిపోయింది. ఎపిడర్మిస్ ఏంటి? outermost layer. And if you move on to this cortex consists of 3 layers, that is an exodermis, general cortex and endodermis in Japan. That exodermis is also called as an hypodermis. It is also called as an hypodermis. There will be another thing called as a general cortex, and there will be last one, that is an endodermis. Okay, so if you move on to this hypodermis is made up of 3 to 5 layers of collenchyma cells which provides mechanical strength, that is flexibility and rigidity ni provide cheyadaniki ee layer anedi upayogapaadutundi. It is made up of 3 to 5 layers of collenchyma cells. Okay, and next it also helps in, next we'll move on to the general cortex. It is a 5 to 10 layered parenchyma cells tho made up ayi. 5 to 10 layers of it is made up of 5 to 10 layers of the parenchyma cells. Okay, So, and this, the main function of this is it consists of a chloroplast as well as a leucoplast. Outer cortex or outer layer of this general cortex consists of a chloroplast and inner layer consists of a leucoplast, that is a colored or colorless substances. Okay, and this helps, this because it has chloroplast, it helps, the main function is it helps in photosynthesis, and it also helps in the process of storage of food materials. And coming to endodermis, it is a uniceriate layer which consists of which is made up of a lignin and suberin bands, and inside its layer there are certain bands or strips called Casparian bands in it. And next, if you move on to the stele, that is the third one. This stele is larger than the cortex and it consists of obviously 4 parts. ఏంటివి స్టీల్లో ఉండాల్సిన 4 parts? Pericycle, vascular bundles, medulla or pith, and this is also called as medullary rays. Or medullary rays. So if you move on with this, first is the pericycle. Pericycle is a discontinuous multiseriate layer present as a bundle corpse, and this pericycle consists or made up of a dead scleral chymatose cells. ఇది డెడ్ స్క్లీరల్ కైమటోస్ సెల్స్ తో మేడ్ అప్ అయి ఉంటుంది. And coming to its function, it provides mechanical strength to this steel structure. And next one is the vascular bundles. In these vascular bundles, these are normally wedge shaped. Those are collateral. Collateral అంటే ఏంటి అంటే xylem ఇంకా phloem రెండు side by side ఉంటాయి. So in this dicot stem, vascular bundles are collateral అంటే xylem ఇంకా phloem ఏవైతే ఉన్నాయో అవి సైడ్ బై సైడ్ సేమ్ రేడియస్ పైన ఉంటాయి. అండ్ conjoint is nothing but joined together. Xylem ఇంకా phloem రెండు ఒక దగ్గరికి జాయిన్ అయి ఉంటాయి. అండ్ open is nothing but the cambium is present. Cambium ఉంటే open అని అంటాం. And endark, and these are endark. Endark is nothing but protoxylem which is present. That protoxylem is facing towards an inner side and metaxylem is facing towards an outer side. And here in this case the stele is described as a U-stele. ఇందులో ఉన్న ఈ స్టీల్ ని మనం ఏమంటున్నాము? U-stele అని అంటున్నాము. Why we are calling it as a U-stele? Because the vascular bundles are arranged in the form of a ring. ఇప్పుడు ఇది రింగ్ అనుకోండి, vascular bundles ఇలా సైడ్ బై సైడ్ ఇలా అరేంజ్ అయి ఉంటాయి ఒక రింగ్ లాగా. సో అందుకే మనం అలా అరేంజ్ అయినందువలన మనం దాన్ని U-stele అని అంటున్నాం. ఓకే, అండ్ medulla, and next one is medulla and pith. This medulla and pith is made up of a parenchymatous cells. And coming to the last, that is a medullary rays. Parenchymatous cells between a vascular bundles. ఈ vascular bundles కి మిడిల్ లో ఉన్న parenchymatous cells తో మేడ్ అప్ అయినవే ఈ medullary rays. So this is all about the dicot stem. Okay, and next we will be moving on. First I will show the diagram of this dicot stem and then we will be moving with our monocot stem. So if you see this diagram, this is a dicot stem. So this is dicot stem ని ట్రాన్స్ఫర్ సెక్షన్లో కట్ చేసినప్పుడు మనకి ఇలా కనిపిస్తుంది. అండ్ దిస్ అవుటర్ లేయర్, దిస్ లేయర్ ఈస్ కాల్డ్ యాజ్ అన్ ఎపిడర్మిస్ అండ్ దిస్ హెయిర్ లైక్ ప్రొజెక్షన్స్ అవుట్వర్డ్స్ టు ది స్ట్రక్చర్స్ ఆర్ కాల్డ్ యాజ్ అన్ ఎపిడర్మల్ హెడ్స్. అండ్ ఈ ఎపిడర్మిస్కి బయట మనకి క్యూటికల్ లేయర్ అనేది ఉంటుంది. అండ్ దిస్ క్యూటికల్ లేయర్ అనేది మనకి ఇలా ట్రాన్స్ఫర్ సెక్షన్ చేసినప్పుడు కనిపించదు. ఓకే, అండ్ అపార్ట్ ఫ్రమ్ దిస్ లేయర్ ఈ ఎపిడర్మిస్కి లోపల మనకి ఇంకొక లేయర్ ఉంటుంది. దిస్ డాటర్ లైక్ స్ట్రక్చర్స్ వేర్ ఐ హ్యావ్ డ్రాన్ హియర్, దిస్ డాటర్ లైక్ స్ట్రక్చర్స్ ఆర్ కాల్డ్ యాజ్ అన్ hypodermis. Okay, and next if you move on to the inner side cells, See, this is the layer which is made up of a parenchyma cells, and epidermis కి లోపల మళ్ళీ మనకి ఇంకొక లేయర్ ఉంటుంది, దాన్నే మనం ఎండోడర్మిస్ అని అంటున్నాం. దిస్ లేయర్ ఈజ్ అన్ ఎండోడర్మిస్ లేయర్, and this is a cycle, and this cycle means this is a stele. Epidermis, this total is a cortex, and this is a stele. So inside this stele we'll be having one pericycle. So ikkada pericycle anedi untundi, and this pericycle consists of some vascular bundles, that is nothing but a stele, and here this vascular bundle xylem and phloem. If you see here, the xylem and phloem has been separated by a cambium. So cambium undi kabatti it is said to be an open condition. Okay, vascular bundle side by side same radius paina unnai kabatti it is a conjoint. Collateral ante xylem inka phloem oke daggara join ayi unnai kabatti. Okay, and the central part is medulla. If you see its transverse section, it appears in this way. See, epidermis, outer cuticle layer, these are the hair-like appendages that protrude outward. Those are called as the trichomes or multicellular layers. And after epidermis, there will be two series of layers that are made up of collenchyma cells, and then 3 to 5 layers of parenchyma cells, and then endodermis. There will be medulla pericycle. This is the phloem. This phloem is separated by xylem by a cambium. Protoxylem is facing towards inner, and meta is towards the outer. So its condition is said to be an endark, and there is a presence of a pit. So this is all about the dicot stem. Now if you move on with the monocot stem, yeah, this is the structure of the monocot stem. In this monocot stem, monocot stem appear in this way. The outer layer, this outer layer is called as a epidermis, and the layer inside to this epidermis is called as an hypodermis, and the vascular bundles, these are arranged or scattered in the form of a ring. Oka ring lopala vascular bundles anni kuda ila arrange ayi undadam jarugutundi. And next if we move on to the monocot stem transverse section, this monocot stem transverse section appears in this way. See, this is an epidermal cells, okay, and there will be an outer cuticle layer, hypodermis. These are the vascular bundles. Vascular bundles lo phloem, xylem untundi, and there will be ground tissue. And if you see the arrangement of this monocot stem shows, an epidermis layer, hypodermis, ground tissue, as well as the vascular bundle. So if you take out that first one is an epidermis, as I have said, epidermis is the outermost uniseriate layer which is rectangular in cells, and the cells are compactly arranged that provide protection to it. And cuticle is a waxy coating layer which is made up of a cutin, and it checks the rate of transpiration. And in the epidermis layer there will be a presence of a stomata ఉన్నందువలన అవి ట్రాన్స్పిరేషన్కి ఉపయోగపడతాయి. And next layer, as I have said, it is an hypodermis. It is made up of 3 to 4 layers of sclerenchymatose dead cells. So ఇవి ఏం చేస్తాయంటే ఆ యొక్క సెల్కి మెకానికల్ స్ట్రెంగ్త్ని ప్రొవైడ్ చేస్తాయి. ఓకే, అండ్ నెక్స్ట్ ఇఫ్ యూ సీ ద ground tissue consists of parenchymatose cells in which numerous vascular bundles are present, and stele kuda untundi, and the stele in the monocots is called as atactostele. నార్మల్గా డైకాట్స్లోకి వచ్చేసరికి స్టీల్ ఏమన్నాము, we have called that stele as a U-stele, whereas in case of this monocot stem, stele is called as an atactostele అని అంటున్నాం. Okay, the stele which consists of numerous vascular bundles that are scattered or irregularly arranged అయి ఉంటే దాన్ని మనం atactostele అని అంటున్నాం. ఎందుకంటే ఇక్కడ చూడండి vascular bundles ఇక్కడొకటి అలా డిఫరెంట్గా స్కాటర్డ్ అయి ఉన్నాయి. Irregularly they are arranged. Because of that we are calling it as an atactostele. And coming to the vascular bundles, if you see, this is a vascular bundle, xylem, phloem, metaxylem and protoxylem. And if the vascular bundles are fibrovascular bundles, those are oval shaped, and those are collateral, conjoined, closed. And so here their cambium is absent. Because of the cambium is absent, these are said to be closed in condition. And vascular bundles in monocot stem are described as fibrovascular, oval shaped, collateral, conjoined, closed, and endark in position. So this is all about the monocot stem as well as the dicot stem. So if you see about this, till now we have discussed about the monocot stem as well as the dicot stem. So in the previous class, that is in yesterday's class, we described about the dicot root as well as the monocot root. And now here we are discussing about dicot stem as well as monocot stem. Rendu kuda same similar gaane untayi. And if you take this monocot stem, as I have discussed now, monocot stem consists of 4 parts. As it consists of 3 parts, it consists of 4 parts. That is an epidermis, hypodermis, cortex, and stem. So now we will discuss about some sort of a differences. And when you are writing an answer, the diagram is very important. So you need to draw the diagram of a transverse section, and you need to observe or you need to draw a diagram like what you have seen under the microscope, what types of tissues or what types of layers that you are being recognized by using this stem. So first, if you are writing about the differences between this, differences between a monocot stem and dicot stem. Monocot stem as well as dicot stem. If you write them or if you know about the differences, in this dicot stem, the trichomes are present. So, quite opposite untundi. Here there will be trichomes are absent. Trichomes absent. Here they are present. Okay. And next, if you see in this case, hypodermis is made up of sclerenchymatous tissue here. Hypodermis is made up of sclerenchyma. And here hypodermis is made up of collenchyma. Okay. And next, endodermis, pericycle are absent here. Endodermis and pericycle are absent. Here they are present. Okay. General cortex is absent, present. ఇందులో జనరల్ కార్టెక్స్ అనేటువంటిది అలాంటిది ఏమి ఉండదు. సో హియర్ జనరల్ కార్టెక్స్ ఇస్ అబ్సెంట్ అండ్ హియర్ ఇన్ దిస్ కేస్ ఆఫ్ ఎ డైకాట్ స్టెమ్ జనరల్ కార్టెక్స్ ఈస్ ప్రెసెంట్. ఓకే. అండ్ నెక్స్ట్ ఇఫ్ యూ సీ గ్రౌండ్ టిష్యూ హియర్ ఈస్ ప్రెసెంట్ అండ్ గ్రౌండ్ టిష్యూ ఈస్ అబ్సెంట్ ఇన్ కేస్ ఆఫ్ ఎ డైకాట్ స్టెమ్. And vascular bundles are arranged in a numerous manner here. This is an important point. Vascular bundles are arranged in irregular pattern. Irregular pattern called atactostele. And here the vascular bundles are arranged in the form of ring. In the form of ring called stick. U-stick. Okay. And next, if we move on, here the vascular bundles are oval shaped. Here those are wedge shaped. Vascular bundles are made up of a fibrous sheet, and vascular bundles here they are not made up of a fibrous sheet. So these are some of the important points and differences in the kachitanga question vastudi. Maybe it may be of root, it may be in case of root, or it may be in case of a stem. And next thing, vascular bundles here are closed. Vascular bundles are closed అంటే ఏంటి, ఇక్కడ క్యాంబియం అనేది లేదు. Here vascular bundles are open అంటే cambium is present అని అర్థం. Okay, and xylem vessels are few in number here. Xylem vessels are more in number there. Protoxylem lacunae is present. Protoxylem lacunae is absent. Medullary rays absent. Medullary rays are present. Pith cavities are present. Pith cavities are absent. ఇందులో ఉన్న వెజల్స్ అన్నీ కూడా వై షేప్డ్గా ఉంటాయి. Vessels are Y-shaped. Here the vessels are in a serial order. And phloem parenchyma is absent in this case, and here phloem parenchyma is present. Stomata is absent in this. Stomata are present in this epidermis. Stomata are absent in the epidermal layer of this. And cuticle once check the rate of transpiration. Here the cuticle and trichomes also check the rate of transpiration. So altogether now I have discussed around about 17 differences between a dicot stem as well as the monocot stem. So in the previous class and today's class, this is an important question regarding a monocot root or dicot root, dicot stem or monocot stem. And the important question comes for short is differences between a dicot root and monocot root, and also the differences between a dicot stem and a monocot stem. And in the next class we will discuss about the dorsiventral leaf and what are the some internal characters of the structures. Okay.